Hello, you guys. Welcome back. We're going to do a little quick review on our genetics chapter. All right. So remember that the genome is the sum of the total genetic material in an organism, and that's even going to include extra chromosomal DNA. So it's not only the chromosomes themselves. Okay. Chromosomes, what are they? They are discrete units of DNA. So they're cellular structures that contain genetic material. In eukaryotes, these tend to be linear, and we tend to have more than one. Archaea and bacteria tend to have one circular chromosome, but they can have more than one, but typically one. All right, your genotype is your genetic makeup. So whatever gene you are carrying from your mom and gene from your dad, but maybe you're only expressing one of those, the one that's getting expressed or what we would see in the world from that result, that is your phenotype. All right, your DNA, we talked about the structure of DNA. DNA is deoxyribonucleic acid. It forms our chromosomes and our genetic makeup. The building block for DNA is the nucleotide, and that consists of a nitrogenous base. So that's what makes it the A, the T, the G, or the C. A deoxyribose sugar, so 5-carbon sugar, it's a pentose. Remember, deoxyribose for DNA. And then the phosphate group. And then that's the basic building block. Repeating, those are nucleotides that would repeat over and over again. DNA consists of 2 anti-parallel strands that run in opposite directions. Remember, when we're talking about anti-parallel, it's essentially like this. You have one going in one direction and the other going in the other direction, right? And then they match up across the way from one another. And one is a coding strand, and one of them is the template strand whenever we are talking about transcription, which we'll get into in a moment. Remind you anyways, right? This is definitely not your first time watching a genetics lecture, right? So anyways, yes, so these always are going to be oriented in the 5' to 3' direction from start. And then the other one's going in the opposite direction, 5' to 3'. So that's anti-parallel. That's what that means. 5' and 3', just talking about the carbon number in that 5-carbon sugar, that deoxyribose, the one that's at the end, that's open at that end. That's all that's talking about. So that's anti-parallel. Replication is semi-conservative. We have the original, that is like the original daddies that will... Sorry, I can't stop laughing when I said that. Sorry. So, anyways, you have the original template strands. So you have the original parent strands, and they're going to split apart, and then we're going to make a copy of the first parent strand. So that's its own thing. Now we have 2 double strands over there, and then a copy of this other strand. Now we have 2 strands over here. So we have one original and one original and one new and one new. Semi-conservative, they each have an old and they each have a new. That's what that means. Okay, so I made it. That's how replication is going to occur whenever we are making copies of DNA, whether we're making new cells or whatever we're doing by making a copy of that DNA. Typically making new cells. So replication for DNA, we have DNA polymerase. It only works by adding in the 5' to 3' direction. So we do need primers that, because they can't add on to nothing. So we do have to have RNA primers that will start everything off. And then once we get that going, 5' to 3' directions, no problem at all. We just go straight 5' to 3'. But remember, we have the 2 strands that are going in the opposite directions. Whenever you split those guys apart, one can easily go 5' to 3'. That's the leading strand. It just keeps going and going. But as the other side is opening up, you can't go into it on the other side because you can only go 5' to 3'. So as it opens up, we'll add on a piece. And as it opens up further, we'll add on a piece, and so on and so forth. And then link all those together with ligase, essentially, to seal it all together. That seals those phosphodiester bonds between the nucleotides back together. All right. RNA. So moving on. So that's the basic rundown of DNA replication. RNA is ribonucleic acid. We have a lot of uses for RNA, right? We have the transfer RNA that's going to carry our amino acids during translation. We have the ribosomal RNA that's part of the ribosome for use. That's the machinery essentially that drives translation. We have the messenger RNA that carries the message of the gene that's going to code for the protein. We're going to read it in threes that we call codons. And then we have like microRNAs and silencing RNAs and all these other things that are used for regulation of expression, less important than what I wanted you guys to know, which is the tRNA, the mRNA, and the rRNA. Those are the big three. All right. RNA consists of nitrogenous bases, but in this case, we don't have ATCG. In the case of RNA, we have a UCG. So A will bind with U and C will bind with G. Anyways, so that's our nitrogenous base. Then we have a ribose sugar, which is a 5-carbon sugar, but it's not deoxyribose, right? It's ribose, it's proper ribose. And then we have phosphates. And yeah, that's our nucleotides that make up our RNA added on to each other, right? Side to side. Okay. Right. So transcription. You're trying to go from DNA to RNA. That's called transcription. Whenever we're doing that, we have 3 basic steps that are involved in that. We have initiation, elongation, and termination. Those are the 3 steps that are involved in transcription. So initiation at the actual promoter region where the RNA polymerase is going to bind onto, and then literally it's just 5' to 3' direction, adding on nucleotides to make the messenger RNA until we get to a stopping point and we disassociate from that. And now we've made our messenger RNA. Messenger RNA in eukaryotes, which are us, messenger RNA in eukaryotes, that's going to have introns and exons. Exons are the coding portion. Introns, we're going to cut those out via spliceosomes. Yeah, they're going to splice. That's what they're going to do. And we're going to cut those introns out. It's important to remember this because whenever you are doing genetic engineering and you're going to be working with bacteria, if you want to put human genes into bacteria and you want your protein made, bacteria cannot spliceosome. They cannot do. So you have to have already taken care of that problem before you put your gene into the bacteria. So that's important to remember that. Okay. Anyways. So DNA nucleotides, A, T, C, G, right? Every 3 is called a triplet. In mRNA, the corresponding mRNA, which is made from the template strand of the DNA whenever we're making it, the 3 that we were talking about in the DNA, they're not triplets now. They're called codons in messenger RNA. So in messenger RNA, we have our 3 nucleotides, and those are going to end up coding for amino acids. Those are read according to the complement of your codon, like A, and a opposite match, the anticodon on the transfer RNA. So AAA would be UUU on the anticodon. And when those match up, that's how the ribosome knows that this amino acid is going to link on to the continuing chain, the growing chain that you're making of your protein, adding on amino acids to amino acids. That process is called translation, and it has the same 3 steps as the transcription had: initiation, elongation, and termination. So we're adding on these amino acids based on that codon-anticodon matchup while we're moving along in the ribosome, and making peptide bonds between these amino acids to make a polypeptide. And cool, so we're making that during translation. How do we know we got to the end? Well, there are specific codons that are nonsense codons that do not code for amino acids, and they cause dissociation of the ribosome from this whole complex. And those are the stop codons, and they don't code for amino acids. Like I said, that's important to remember as well. Right. So that's how translation works. Let me see if there's something else that I'm missing. We did talk about in the lecture too how COVID-19 vaccines work, basically by putting messenger RNA into your muscle cells. Your muscle cells will read that, translate it, and put the protein of the actual virus that was coded in that messenger RNA onto their own surface of their own cells, and they'll put that out there, and then your immune system will react to it. This is normal stuff. This is what your body does when it sees something foreign. So yeah, so it's going to put it on its little surface and display it so that your immune system can come along and be like, that's not right, and make antibodies against it, so that the next time that you see COVID, hopefully you have protection. So that's how that is supposed to work. Of course, viruses mutate really fast, and so it's hard to keep up with that, but we already know that from flu. So okay, the LAC and the arginine operons, these are going to be inducible and repressible operons, respectively. So let's go start with the arginine one first. That is our repressible one. So we're always making arginine, not us. Okay, I want to be very clear about this. Only bacteria do this. Bacteria are always making arginine. And so when they have too much of it, the too much arginine builds up to where it can associate with the repressor protein. And when it does bind to the repressor protein, that tells the cell we have too much arginine. The repressor binds onto the operator. The RNA polymerase can't make any more messenger RNA now because we are blocking transcription of that gene. So that's basically how that's going to be working, physical blocking. The other one is the LAC operon. This is inducible. These genes are not always on. Instead, when lactose is present, we can turn on these genes by the lactose. Normally, at the operator, repressor is sitting on the actual repressor sitting on the operator, like let's say like a troll, like blocking like the bridge or whatever. And lactose comes along and it like feeds the troll. And so now the troll runs off and chases after lactose or something. And now the RNA polymerase can start making messenger RNA. That's only whenever lactose is present that we will be making the genes associated with breaking down lactose. So that's what the LAC operon is and how that works and how it's inducible. We induce the production of those genes that are associated with that. Horizontal gene transfer. The three biggies are going to be conjugation, transformation, and transduction. Essentially, this conjugation is movement of a DNA between bacterial cells via the sex pilus, so making copies and just feeding it through to the other cell. Transformation is picking up DNA from your outside environment. If you are a competent cell, you can do that. And then transduction is whenever you get infected with a bacteriophage that is able to undergo, that is a temperate phage. We call them temperate phages. But if they insert their genome into the host genome that they're infecting that cell, then that is going to be called a prophage, and that is lysogeny. And so when that happens, they can take parts of the genome with them when they leave. So that's specialized. You also have generalized transduction, which just means that like you're building up a virus particle and taking up the little parts of the virus particle, putting your virus together, and you're packaging up bits of the DNA from the host too with it just randomly. So that can lead to transferring genes to new bacteria that way. So that's sort of random though, but it can happen. Then we talked about mutations. We talked about the mutations, the missense ones, where we're going to have a change in the amino acid. So one nucleotide gets changed, leads to a change in the amino acid. That's missense. If we had, however, actually changed nucleotide and we have changed from, we did code for amino acid, but now we have a stop codon there. We have no amino acid there where there should be. That is a nonsense mutation. So that's the stop codon one. If you have a change in your nucleotide sequence, but you're lucky enough that it falls within that wobble safety area, then that's going to be a silent mutation where you have a change in your nucleotide sequence, but not in your amino acid sequence. So same amino acid, even though you changed the nucleotide. Frame shift mutations, we have the actual reading frame. We have like read these three, these three, these three. And that's the amino acids that you'll get by reading those specific three. If you take out one of those nucleotides and shift everything over, suddenly that same three in that frame doesn't read for the same amino acids. And that's a frame shift mutation. So these can be very severe and very, very detrimental to the organisms. So that's the basics of microbial genetics. Yeah, for sure. And we'll see you guys for the next one.